Hey everyone, and welcome. Today, we are taking a look at FragPipe, a complete proteomics pipeline that's built around the super-fast MSFragger search engine. If you're working with mass spectrometry data, you're definitely going to want to see this.
You know that feeling, right? You've run all your samples, and now you're just staring at this mountain of complex data. It can feel like a huge leap to get from those raw files to real biological answers.
Well, there is a better way. FragPipe is an all-in-one platform with a graphical interface that's designed to walk you through that whole journey, from the moment you load your raw files to the second you get those beautiful outputs ready for analysis.
Okay, let's jump right in. The very first place we're going to go is what I like to call Mission Control, the Config tab. This is ground zero. It's where we make sure all the powerful tools that FragPipe uses are connected and ready to go.
Getting set up is actually pretty simple. First, you'll hit the download/update buttons to grab the latest versions of the core tools. Then, you just have to tell FragPipe where to find heat programs like the MSFragger search engine and IonQuant for quantification. And finally, you've got the option to link a Python installation, which opens up some even more advanced features later on.
And here's what it looks like when you're all set. See, this Config tab is your central dashboard. You link everything up just one time, and from then on, FragPipe knows exactly where to find all its engines for every analysis you run. It's a quick, one-time setup that puts some serious analytical power at your fingertips.
All right, with our tools all configured, we're ready for the fun part. Let's head over to the Workflow tab. This is where we get to design the actual blueprint for our analysis. And this is just so important. Think of the Workflow tab as the place where you tell FragPipe the whole story of your experiment. Having a really clear plan here is the secret to getting results that are not just accurate, but also meaningful. Now, one of the best things here is that you don't have to start from a blank slate. FragPipe comes loaded with pre-configured workflows for all the common experiment types. So, for example, we can just pick a standard workflow for data-independent acquisition, or DIA. It's the absolute best way to get up and running in no time.
So we've got our blueprint. Now it's time to bring in the data. You just click Add Files, and you can load your raw mass spec files. And FragPipe is super flexible. It handles standard formats like MZML, but it can also work directly with vendor files from Thermo and Bruker.
Okay, this next step is absolutely crucial if you want to get meaningful results. You have to annotate your files. That just means giving each one an experiment label, like control or treatment, and then a bioreplicate number. It seems simple, but this is what lets FragPipe know how to group your data for all the downstream statistical tests. And the last piece of the puzzle is the protein sequence database. You can think of this as the protein dictionary that MSFragger is going to use to identify all the peptides in your sample. FragPipe makes this so easy. You can download a top-quality database right from Uniprot. It even automatically adds in decoys and common contaminants, which are essential for making sure we can trust our results.
All right, the setup is done, we've got the blueprint, the data is loaded, and our protein dictionary is ready. So, what actually happens when we hit that Run button? Let's take a quick look under the hood.
The moment you hit Run, FragPipe kicks off this whole seamless pipeline. First, it uses the super-fast MSFragger to search for matches between your data and that protein dictionary. Then, it validates all those matches to really boost our confidence. After that, it does protein inference, which is basically just figuring out which proteins are really in your sample. And finally, it quantifies everything. It's a completely automated end-to-end process.
And this is where things get really, really smart. FragPipe uses some seriously advanced tools. For example, MS Booster uses deep learning to predict what your peptides should look like. Then, it hands those predictions over to another tool called Percolator, which uses that extra info to get way better at telling good identifications from bad ones. The result? More confident IDs from your data. So running one analysis is cool and all, but let's be real, modern proteomics is all about scale. What if you've got dozens or even hundreds of files to run? Well, that's where FragPipe's batch mode is about to become your new best friend.
So the basic building block of batch mode is something called a job. And a job is really simple. It's just one complete FragPipe run. Your workflow, your files, your settings, all bundled up and saved into a single file.
And here's the magic. You can create a whole bunch of these jobs, line them all up in the batch queue, and just tell FragPipe to run them one after another, completely automatically. This is how you really scale up your research and save a massive amount of time. And making a job is super easy. You only need 3 things: your saved workflow file, a manifest file that lists all your data, and a folder where you want the results to end up. That's it.
Okay, after all of that setup and all that processing, we finally get to the best part, the payoff. It's time to explore the results and see what kind of data FragPipe has for us.
Because that's the whole point of all this, isn't it? We're not just trying to make tables full of numbers. We're trying to get to organized, quantitative data that can lead us to some exciting new discoveries.
One of the most important files you'll get is the protein matrix file. It's just a text file, but inside you'll find your protein IDs, gene names, and, most importantly, the quantified intensities for every protein across every single one of your samples. This is where your biological story really starts to come into focus.
But you are not stuck just looking at spreadsheets. Right from the Run tab, you can fire up the FragPipe PDV viewer. This is an awesome integrated tool that lets you dig so much deeper. You can visually inspect your peptide IDs, look at the actual spectra, and really confirm your most interesting hits with your own eyes. At the end of the day, all those answers you're looking for are locked away inside your complex data. A tool like FragPipe is just there to give you a powerful and really friendly way to find them. So the only question left is, are you ready?
If you want to give it a try, you can download the software and find lots of great tutorials over at fragpipe.nesvilab.org. Thanks so much for tuning in.